in food production, and what we said is we have just started the introduction part of that chapter and we have talked something about plant breeding, right? What we remember from the last class, we have talked about certain strategies, certain techniques that are adopted for enhancing the food quantity and also the quality of the various crops, right? And why do we have to enhance the food, or why do we have to increase the food? One basic reason is because of the growing population. This is something that we have talked about, right? As far as the various techniques are concerned for improving the quantity or the quality, under that heading we need to talk about certain techniques, and the first that we have started was plant breeding. What is plant breeding? How is the process done? This is the part that we have already completed, right? Next is together with plant breeding, right? Since why we are going for this breeding program? We are going for this breeding program so that we can enhance the food, right? Food to jate ami besi koribo paru. And when we are talking about this, we have to talk about another thing that is called as green revolution, right? Now what exactly is green revolution? Green revolution is nothing but a period, no, at a specific years or at a period. Hey period to ki hoisile? Hey period to there were certain crops that got tremendously increased, right, by the incorporation of certain advanced technology, certain advanced seeds, certain advanced agricultural practices, and that period is called as green revolution. Talking about green revolution, what you need to study within this: first, what is green revolution? It is a period. Second, when we talk about green revolution, basically two crops we talk about, that is increase in rice and wheat, and to be precise, we talk about something called as a semi-dwarf variety of wheat and semi-dwarf variety of rice. After that, another important question that is asked is about the father of green revolution, Indian father of green revolution and the normal father of green revolution, right? So, hekini poribo lagibo. And we often talk about semi-dwarf variety, right? Semi-dwarf variety of wheat, or if I ask who is responsible or what is responsible for the green revolution or for the enhancement of food during that period, is something called as semi-dwarf variety of wheat and semi-dwarf variety of rice. Now what we understand by the term semi-dwarf variety, wheat and rice, obviously we know that these are nothing but two major crops that are found in India, and the entire population is dependent on these two major food, right? Semi-dwarf variety mane ki? What is semi-dwarf? Dwarf is something that we understand. What is dwarf? A very short form of organism, that is what is called a dwarf. Semi-dwarf, neither too long, neither too tall, neither too short. The middle stage is something that is called as semi-dwarf, right? Basically, ukho nohoy, jiman hobo lage himanu nohoy, ekebare dwarfu nohoy, major stage jok ami ki boli kau semi-dwarf. Now, in order to make a plant semi-dwarf, first though, why are we using the word semi-dwarf? Because scientists have proved it experiments that if the plant was made semi-dwarf, the quantity, the quality of that particular plant increased. So, if you are talking about a wheat plant, normal wheat plant edale jiman kini production dibo, semi-dwarf variety production to it will be more, the quality would also be much better, right? So, normal semi-dwarf mane as compared to the normal height of the plant, that ke eta reduced form. Eta plant of reduced form koribo lagile, if you want to make a plant semi-dwarf, what do you need to do? Obviously, you have to go for breeding. Breeding mane ki asile? Changing of traits. So, what exactly you are doing is, you are doing something with the gene, right? So, adult plantain, what we do, we incorporate certain types of genes, and ultimately because of that gene, ki hoy plant dal semi-dwarf hoy. Semi-dwarf hole what we have seen is the quantity, the quality has improved according to the gene that we have incorporated into the plant. Amak bhal quantity lage, gene to belek hobo. We want good quality, we want good color, we want good protein content, etc., etc. Accordingly, ami ki korim, we'll be incorporating certain types of genes for that. So, this is what is called as semi-dwarf. Next is semi-dwarf. Actually the entire process started in, or green revolution started in, first the year. We generally we are not precise with the exact year, 1965 or 1968 boli ami nokoi pelai. In general what we say is green revolution is something that has started during the 1960s. Right? So, teneke question ahibo. So, we will be concerned with the word 1960s. Our tatkeo jodi precisely ahe, it is 1965 that the question is asked about, because that is something that has started in India, right? As far as worldwide is concerned, green revolution code start hoise, Mexico start hoise, right? And we have to talk about the father, Norman Borlaug, right? And these are the questions that we will be talking about, right? So, please go through the notes. Since we have already given you the PDF, we do not have to write anything, just go through. Yeah, please say. Ask. class 2 kihor soli ase. This is a class about our new topic, right? So if you have doubts regarding any question, regarding any test, it is always after the class that you ask, right? Class 2 complete koribo laibo. After that only you have to ask me the doubts, not within the class. Okay? Class 2 hoilok, class 2 hoilok, tarpsot tumi muk doubts koriba. Hobo? A lesson to complete kori lo. Okay. Okay. Okay, just see. Now, what are the points that are important from these notes? Okay. First is Green Revolution. What we understand? It is a period started in 1960, right? And the Green Revolution. What was that? What got revolutionized? Key change hoisile. It was the incorporation of. Have you heard something called as high yielding variety? High yielding variety to know HYV seeds, right? So it was the introduction of all these good varieties of seeds, good agricultural practices, good tractors were incorporated. Everything led to the industrialization of the agricultural field, and as a result the increased quantity was the result, and so we got much high amount of food as compared to the previous, right? As we are comparing, yate duta point ase, so something about rice and something wheat. So from 1960 to 2000, wheat production increased from this to that, and rice production increased from this to that. The question is asked about that. So wheat kihor pra kihole increase korile 11 million to 75 million, and if you are talking about rice, it's given there 35 to 89.5. So these two facts yar upor question ahe, ekita important hoy, thik ase. Next is obviously India is an agricultural country and all ebilak to ami janu. Agriculture contributes about 33%, again 33% about question ahibo pare. Next, father of Green Revolution, just come down, see the points. Indian father, Swaminathan. Generally the question has been asked about the Indian father only. ठीक है सर, Norman Borlaug और ऊपर इमान question आहा नहीं. Swaminathan is something that you need to remember. There is a question that follows, just read the question. What is responsible for enhancement in food production during Green Revolution? Means you are asked to name the two crops which helped in increasing the quantity of the food. It is obviously wheat and rice, right? Okay. So which form of wheat, which form of rice? Semi-dwarf variety of wheat and semi-dwarf variety of rice. Talking about semi-dwarf, obviously there was a particular gene in the plant and you have to know the name of the gene. If you are talking about the semi-dwarf wheat, kuntu gene responsible asile? We talk about a gene that is called as Norin 10. ठीक है? So asan tar likha ase. Dwarf wheat buli likhat first point to ki diya ase? Dwarfing gene, right? The gene that was responsible for making the plant dwarf, it's Norin 10, and this gene was first reported in Japan, right? So baki kita points porha. So it's only the name of the gene that's important. Baki duta points of our question aha nai. Okay, next what is important is the two names. Variety of wheat ketaman diya ase na, Sonara 64, Larma Roja 64, Sonalika and Kalyan Sona. Question keneke ahe? Identify the variety. The question might be asked, Kalyan Sona and Sonalika are varieties of rice, wheat, barley, sugarcane, etc. Right? Next is Green Revolution or Karne, jintu incorporate korisil ba jintu introduce korisil India le. What are the varieties? It is Sonara 64, Larma. Actually, it is not only these two varieties that were introduced in India. First, এনেকুৱা ধৰণৰ plants বিলাক বা experiment বিলাক ক'ত কৰিছিল? Mexicoত কৰিছিল, because we know Green Revolution started in Mexico, right? Later when it was seen that these had fruitful effects, they were later incorporated in India or introduced in India. প্ৰথমতে Mexicoত হৈছিলে, তাৰপিছত ইণ্ডিয়ালৈ আনি পেলাই Indian climateত ৰুইছিলে, ঠিক আছে? So when we talk about varieties of wheat that were brought from Mexico to India, generally we have to study about two of them, that is Sonara 64 aru Larma Roja 64, but it is not only these two that were brought, it was almost about 5 varieties of wheat that were brought from Mexico and was introduced in India. ঠিক আছে? কিন্তু পাঁচটাৰ ভিতৰত question নাহে, এইকেইটা আমি পঢ়িম. It is only the names that you need to remember. Next, okay, Sonara 64, Larma Roja 64, just read the line. They were brought to India and modified. Sonara 64, Larma Roja 64 ki hoy? Dwarf wheat variety. Code develop korisile, Mexico develop korisile. They were the two crops that were grown in Mexico properly. If you want to incorporate or introduce one foreign crop into your own country, sometimes it might be like that because of the prevailing environmental condition, that type of a crop might not grow in India. So it calls for certain modification in the plant. Mexico due to environmental conditions asile, obviously India to hetu nohoy. So the same plant cannot be growing at the same extent in India itself. So it got modified. Question ahe, modified by what? Gamma modification. Gamma ray di pele modify korisile. So please underline that word and you have to answer that. Remember that, okay? Modified by gamma mutations. Next is below that. In 1963, many varieties like Sonalika and Kalyan Sona were introduced at all the belts of India. Egita ki hoisile modify kora nai. Agor gita ki hoisile modify korisile, right? Ar modify kihere korisile gamma radiations ba gamma mutations, right? So these are the things. What is important? The name of the gene. Second, the plants that were introduced to India from Mexico, right? Naam gita janibo laibo. Next, modify korisile, kihere modify korisile. Gamma ray. Just these 3 points that you have to remember, right? Okay. Similarly, in case of rice also, we have been using a word called as semi-dwarf variety of rice, and if you have to make a plant or if you have to make the rice plant dwarf, obviously you have to play with the gene, you have to introduce a new gene into the plant. And what is the name of the gene? Again, it's given there. His was a DGOU gene. DGOU gene, that's the name of the gene. And jeneke Norin 10 reported in Japan, A2 code report korise Taiwan, right? Okay. And next information kisuman dia ase Rice Research Institute code ase, International Rice Research Institute code ase, ebilakor report questions ahibo pare. And the last point, semi-dwarf variety of rice developed in India are Jaya and Ratna. So question kya hai? Jaya and Ratna are the varieties of rice, wheat, sugarcane, barley, etc., etc. So you have to know about that, right? Wheat variety, rice variety. So this is all about Green Revolution that you have to study. In a lump sum, what is Green Revolution? A period at which the food quantity increased. Which food to be precise? Rice and wheat. Right, rice or kuntu semi-dwarf variety, wheat or kuntu semi-dwarf. Now, can you say why was the semi-dwarf variety better as compared to the normal one? Ki problem hosile normal one to the semi-dwarf banabo laga hol. Normal rice or wheat plant are at a normal height ase, right? But you have made the plant dwarf or you have shortened the height. What was the reason or what might be the reason behind? Apart from the nutrition, apart from the fruiting, that is something different. Kintu dwarf hole advantage ase na disadvantage hobo. Especially crops like rice, wheat, maize, etc. One basic thing is, as the these are nothing but monocots, right? They don't have secondary growth. They are obviously weak, lean and thin, right? So if they are very tall, there is a chance of lodging, right? What is lodging? Bending down. That is what is called as lodging. And if it bends down, fruit to the matit lagi jai, obviously the fruit will get what? Destroyed. So it's very good that you make the plant semi-dwarf so that lodging problem to no hoy, along with the wind, along with the water, etc., etc. So that was one of the reasons why it was made semi-dwarf, and later it was seen that semi-dwarf hole ki hoy, production besi hoy. Clear? Actually, revolution process to kune korisile. So, green revolution mane okol se rice or wheat, okol se duta planter. Someone is talking at the background, please. Okay, okol se rice or wheat ba okol se duta planter kotha we are concerned enuka kotha nohoy. It is a total period of change. Not only the good varieties of seeds are concerned, but together with that, improved agricultural methods is also a part of green revolution. Okol crop kitai green revolution nohoy. That improved variety, improved agriculture, improved tractors, improved chemicals, improved fertilizers. Gute bilake mili pelai hai tumak ki korile. Introduce ba junkini food production asal tak besi korile, right? Our jihetu we are talking about India, the major food obviously rice and wheat. Our green revolution or maximum experiments was done on rice and wheat. So whenever we talk about green revolution, it is just these two plants that we remember. Clear? তোমাৰ বেকগ্ৰাউণ্ডত কোনোবাই কথা পাতি আছে নি বাৰু Okay, please maintain a little bit of silence, okay? মই বস্তুটো clearly ভালকে শুনা পোৱা নাই, okay? So, তোমাক কি লাগে meaning between dwarf and semi-dwarf? What is your question? Question to সোধা আকৌ তাকেতো কোৱা তুমি questionটো কোৱা ভালকে কোৱা শুনা নাই Dwarf or semi-dwarf or meaning to know. What is the opposite of tall? Tall or opposite ki? শুনিছা নে তুমি তলৰ opposite কি बड़ा छोटा होता है नाटा that is short right okay তলৰ opposite জানা না জানা তুমি উখ মানুহ নহলে কি বুলি কও opposite কি সাপৰ মানুহ হয় নহয় dwarf তোমালকে কি কোৱা বাউনা word চিনি পোৱা বুজি পোৱা dwarf হলে বাঢ়ি না যায় dwarf হলে dwarfে হৈ থাকিব যদি এডাল প্লান্ট নরমালি যিমান উখ হয় ন রাইস এডাল হক হুইট এডাল হক হেবিলাক প্লান্টস কি হয় লিনেন থিন হয় ন মোনোকট হয় প্লাস এডভান্টেজেস ফাইব্রাস রুটস থাকে সো তাহাতৰ কি হয় লজিং প্রবলেম এটা থাকে হোয়াট ইজ লজিং যে বেছি উখ হৈ গলে গছডাল বেকা হৈ যাব ভাঙি যাব আধাত যাব না যায় রাইট হেতুক আমি কি বুলি কও লজিং সো গছডাল তুমি যদি ইমান উখ নকৰাই তেতিয়ালে ভাঙি যোৱাৰ চান্সটো কম থাকে না থাকে সেই কাৰণে গছডালৰ ফ্ৰুটটো ইফ ইউ আৰ টকিং এবাউট ৰাইস অর ইউ আৰ টকিং এবাউট হুইট যোনটো সিডস হয় সিডস বিলাক ক'ত ডেভেলপ কৰে এপেক্স অফ দ্য প্লান্ট হয় নহয় অলমোস্ট গছডালৰ টিপত কৰে নে নকৰে মাকৈ গছটো দেখিছা চাগে ৰাইসো দেখিছা হয় নহয় সো যদি টিপত তোমাৰ ফ্ৰুট বা সিডস খিনি আছে এটো যদি উখ হৈ পিলে ভাঙি যায় তেটা সবতছে হ্যাম্পার্ড কি হ'ব সিডস খিনি হ্যাম্পার্ড হৈ যাব না যায় রাইট সেই কাৰণে যিমান ডুৱাৰ্ফ হয় হিমান ভাঙি যোৱাৰ ক্যাপাসিটি বা টেন্ডেন্সিটো কম হয় সেই কাৰণে প্ল্যান্টডাল ডুৱাৰ্ফ বা সেমি ডুৱাৰ্ফ কৰিছিলে What is the difference between dwarf and semi-dwarf? Dwarf মানে পুরা একদম যিমান ধৰা 10 cm আছিল dwarf বুলি কলে ধৰা 5 cm semi-dwarf কি হ'ব 7 cm বুজি পাইছা not dwarf not tall মাজৰ ষ্টেজটোক আমি কি কৈছো semi-dwarf dwarf আৰু semi-dwarf কৰিলে কি হয় গছডালৰ ভাঙি যোৱা কি টেন্ডেন্সিটো কমি যায় Okay? Understood, na? Okay. So this is all about green revolution. I think it's clear now. Okay. The next topic talks about what? Plant breeding for disease resistance. We are talking about something called as plant breeding. What is plant breeding? Nothing but experiments, right? It is something that we play with the gene and make new plants, right? When we are talking about plant breeding, why do you go for plant breeding? To improve the quality, to improve the quantity, etc., etc. So one characteristic of a plant is we want a plant that is disease resistant, right? Jate niki hunkale bemar hui na jay gosdalot. So we want to make a plant that is disease resistant, and whenever we talk about disease resistant, there are certain factors that depends on edal gosot bemar kiho karne hoy. Why does the plant get diseased? Eta organism disease kiho hai amar bemar kiho karne hoy kisman conditions karne bemar hoy no we have a low resistance so we fall sick similarly in case of a plant also there are certain factors because of which the plant get diseased so we have three conditions we study about them what are the three conditions we talk about the pathogen that infects the plant disease hoise kiho karne kiba ta microorganisms khumaise so who is responsible the pathogen is responsible second pathogen is responsible second the gene of that plant is also responsible you can compare it with human beings also for example common cold and cough je hoy no that's a viral disease everyone is exposed to the same virus but it is only few of the people that get infected and few do not get infected in spite of staying in the same environment what is the reason behind দুটা মানুহ আছে একেটা এনভায়রনমেন্টতে আছে হে এনভায়রনমেন্টতে এটা ভাইরাস আছে হে ভাইরাসটো এটাক এফেক্ট কৰি এটাক যে নাই কৰা কিহ কাৰণে maybe the other person is more resistant, right? Their immune system strong, right? So ultimately disease hobo lagile ketaman conditions upor depend kore. First, it depends on the microorganism or it depends on the pathogen that will enter. Second, it depends on the gene structure of the organism. Amar genetic makeup kenekwa, right? Protective organism or genome upor depend kore. And third is the environmental condition. So these three factors decide whether the organism will be diseased or not. Clear? Okay. So hekinie likha ase. The development of a disease in a plant depends on the interactions among the following factors, that is the host genome. Host genome mane the genetic constituent of the plant or the genetic constituent of the organism. Jar disease hobo, right? The pathogen genotype or the gene of the pathogen and the obviously the environment. Some host baki kita swa. Yata next ki dia ase the definition of resistance aru susceptible. What is the opposite of resistance? You are resistant to a particular virus, you are dash to a particular virus. Opposite of resistant ki? Chemistry है तो यहाँ पे है ना? Yes. Bio में वो नहीं होता है. Susceptible. Susceptible. Okay. So either you are resistant or you are susceptible. So the next two lines talks about what is resistant and what is susceptible. So pori so buji paisan nahi. A host which are not resistant to a pathogen are called as what? Susceptible, right? So susceptible or disease hobo, ne resistant or disease hobo? Resistant or definition to moi repeat nokoru likha ase swa. Pori swa doubt to khuda. Ha? To okay. Okay, so mobile to nai need tumar tat. Oh, beautiful. Beautiful. May I know your name? Naam to question to? Okay, very good. Virat futi no aji sare log ese pori no porile hobo aji.
Ma'am kya naam nikla?
Robial. Hai na? Etia kwa. Swa. Okay. It is not like this that a PDF was given only yesterday. Kiman din agote PDF disu. Tumi aji somoy poa nai, okay, accepted. Baki kidin ki আজি ৰাতি ওলাব মোৰ আজি ক্লাছটো কিন্তু আজি হৈ যাব তোমাক মই কি কৈছিলো হেডা তোমাক বুলি নহয় সবকে কি কৈছিলো যে ক্লাছৰ আগতে পিডিএফ টো ওলাবা আৰু কিমান দিন আগতে দিছিলো অলমোস্ট अराउंड 4 to 5 days আজি হে খাৰু পেটিয়া গৈছা এই 4 দিন তাতে আছিল নি anyway there is no point okay chalo so there are two words প্ৰথমখিনি বুজি পাইছা নে নাই adult plant or disease kiya hoy it depends on three factors right next when we are talking about disease in a plant no adult plant or disease hoise ne nai duta words come to our mind one is resistant the other is susceptible duta words ase resistant to the disease susceptible to the disease resistant mane virus to ahile ahi pelai tumar gar bhitorot bemar tu koribo para nai you are not becoming ill that is what is called as resistant right tumar resistance power besi he karone bemar hua nai tak ami ki boli kom resistant right tumar resistance kom he karone tumar bemar hoise tak ami ki boli kom susceptible is it clear okay next come to the next part a successful breeding for disease resistance depends on ekhini to buji paisu sage just see a good source of resistance dependable disease test etc right now when we are talking about disease and the different plants below this there are certain names of disease and the pathogens that cause it right jeneke kisuman these are very important late blight of potato ebilak disease ami jetia fungi porisu tetiao porisilu if you remember right so at that time also what I said is you have to know the name of the disease and the causative organism kune cause korise is it a bacteria because whenever we talk about disease The credit goes to whom? Obviously the microorganisms, and which microorganisms to be precise? It is bacteria, it could be virus, it could be fungi, right? Or it could be certain types of small animals also in certain cases, certain types of insects also, right? But majority kune disease cause kore microorganisms bilake, microorganisms buli kole bacteria, fungi aru virus, right? So yate kisuman disease ase, disease of causative organism kita mukhastha koribo lagibo. Aru question keneke ahe? Suppose late blight of potato is caused by, it is caused by, name of the pathogens lagibo, Phytophthora infestans, or vice versa. Phytophthora infestans causes late blight of potato, right? Second question regarding this, late blight of potato is a bacterial disease, fungal disease, ne viral disease. Hey bostu to jani bole, it's a fungal disease, right? It's a fungal disease. So this is the way you will read. Ekhinit basic we don't need to explain because we have completed during fungi, right? All the points are very important. Are egita reported question ahe, nothing more than this, right? It is always late blight of potato, loose smut of wheat, black rust of wheat, bacterial blight of rice, cucumber mosaic, and root knot of tomato. A pastar uporte ghurai pokai question to ahe, egitai ami okol porhim. Thik ase? Especially from this chapter. As far as fungi is concerned, rest of the things we need to study. Ekhinir pora kiba doubts ase niki swa? Some important disease. I think it's understood. Swa. Okay, come to number 5, cucumber mosaic disease. Pathogen to swasun tarpot question ahe. First, it is a bacterial, viral, na fungal. It is a viral disease, right? Yeah, and what is the genetic makeup? Whenever we talk about virus, I think you remember, virus buli koa loge loge tar genetic material thakibo na thake, obviously thakibo. Genetic material buli kole what we understand, DNA and RNA. So is it both DNA and RNA present in the virus, or is it either DNA and RNA present in the virus? It is either, no? Both kot thake, mycoplasma, right? Remember we have talked about all this, right? So if you are talking about this one, so it will have either RNA or DNA. RNA generally single stranded and double stranded we have talked about. Yate ki ase single stranded ase. And what we remember about RNA virus, RNA bilakor ki hoy, generally plant disease RNA or generally animal disease DNA, and we had certain exceptions to that also. Okay. Number 5, it is not a bacterial, not a fungal, not a viral. It is something that is caused by a nematode, right? It's a small organism, I mean animal. Okay. Next is, how will you make a plant disease resistance? You can make a plant disease resistance by two methods. Either you can go for conventional method or you can go for mutation breeding. If you are talking about convention method, we are talking about what? All the steps of plant breeding that we had discussed in the last class. Last class or ami plant breeding keneke koribo leke discuss korisu, no? I think you all remember something called as collection, selection, right? Then we have talked about testing, right? Testing of the parents, testing of the individuals. Then ultimately it was commercialization. If you go by this long process, that is what is called as conventional method for breeding, right? Adult plant disease resistant koribo lagile, you can go through this all methods, or you can directly go for mutation breeding. Modern date ki hoy, short bostu to use kore, so we just go for mutation, mutagens kisuman apply kore, will change the gene and ultimately will make the plant disease resistant. Is it clear? Okay, convention method of mutation breeding. Next, talking about this lesson, no, yar bitor maximum questions ki hor upor ahe. There are two charts that's given in your NCERT book. Table 9.1 and table 9.2. If anyone has an NCERT book, please open. Ase no? Table NCERT 9.1 page number kiman? 175. Aru? 175. Right. So in this lesson, very commonly asked question is about these two charts, right? Each and every column of the chart you need to by heart aru upai nai. You have to by heart only. There is nothing to explain, right? The ki dia ase soson name of certain crops, tar varieties bilak infected by what, right? So if you don't have the NCERT, you can see it later, right? It's there. Ekhini khali mukhosto koribo lagibo. Maximum questions has been asked about this, right? Plant breeding for developing resistance to insects. Aru second ki asile some crop varieties resistant to fungi, bacteria and viral disease. Education chart important. Below this, what is given for certain points? Solid stem in wheat leads to non-preference by the stem. Sorry, next second. By a particular fly that is called as sawfly. Question 2 hai, why doesn't a sawfly infect the wheat variety? Because it does not have key. Because the stem is solid. Jedi stem bilak ki hoy, the cells are loosely arranged. You have food material stored within that. If all the cells are compactly arranged, the insect cannot invade the plants and so the stem is not infected, right? So question eta hai, the sawfly kak infect koribo nware. Sawfly solid stem of wheat infect koribo nware. This is the way that we will read the sentence, right? Next, another example, smooth leaved and nectarless cotton varieties do not attract a particular type of a worm that is called as a bollworm. So bollworm e kak infect no kore? Cotton. Which cotton? Nectarless cotton, right? Kunto. No, that's the name of the insect. Bollworm eta insect. Cotton plant alok hetu insect infect nokore. Kyo nokore? Karon nectar nai. Right? Kiba eta khwa bostu pabo lagibo no? Tetehe insect tu sabo. Okay? And another example has been given about maize. Etu question iman important no? Oh yes, stem borer. Stem borer chart khonot ase. We'll study all the examples from here only, right? Oh, this is a chapter that you have to by heart. There's nothing to understand. 9.1, 9.2 totally mukhosto korim. Ar baki eku nai yate. Yeah. Next, we have a topic called as biofortification. Biofortification over to question ahe. Please say. Chart duta mukhosto korile hobo. Not necessarily it's get cotton insect pollinated to no, it's air pollinated. Or no, anemophily he kind of cotton to ki hoy, fluffy hoy, it goes in ki along with the wind. Okay, the next topic is biofortification. What is biofortification? Definition of a question ahe. What is biofortification? It is increasing the quality of a plant. You increase certain chemical compounds of a plant. For example, if you want to increase the protein content of a plant, if you want to increase the amino acid of a plant, right? So tenukwa jeti specific kiba eta bostu increase koribo paru for the sake of humanity, that is what is called as biofortification. So swatat ki likha ase breeding of crops with higher levels of vitamins, minerals, proteins and fat. So you make a plant, hey plant dalot ki korisa normal jiman protein thake tatke besi protein lage amak, he karone ami ki korisu tar gene to change korisu. That is what is called as biofortification. You modify a plant to increase either the vitamin content or the protein content or the mineral content, etc., etc. That is what is called as biofortification. Here biofortification upor question ki hai? First, the definition. Second, so asan duta star di duta point ase. Cereals are commonly deficient in and legumes are commonly deficient in, right? Cereals are commonly deficient in lysine or tryptophan. Legumes are commonly deficient in sulfur containing amino acid, that is cysteine and methionine. Maximum question has been asked about this 2 point only, right? Directly ahibo, legumes are deficient in, or question ahibo, cereals are deficient in. Eta point khali by heart koriba, that's all, right? And since cereals to fat no hoy, cereals can get fat over rice, wheat, etc. Rice, wheat, eblak ki hoy? Examples of cereals. Legumes or example ki? all the different types of dals that we have, pea, bean, gram, eblak ki hoy, legumes, rice, wheat, eblak ki hoy, cereals. Okay, so heblak plant general deficiency ki hot hoy, ekita bostu ami monot rakhim, lysine aru tryptophan, methionine aru cysteine. Next examples has been given about high lysine maize. Tamane you have the normal maize, you have made a plant or you have made a new maize plant with high lysine content. You have changed the gene, you have prepared a plant, hey plant dale as compared to the normal plant amount of lysine basically dibo, right? So you have done the breeding program just to increase the content of lysine. This is called as biofortification. You have done a breeding program just to increase the vitamin content of a plant. This is called as biofortification, right? So accordingly, kisuman examples dia ase high lysine maize, jenenge proteina, ratan, sakti, etc. etc. Swa egita swa example kita swa. Examples of what basic question aha nai. From biofortification part, it is just a definition and the two sentences that we have said. Legumes are deficient in and cereals are deficient in. Biofortification understood or did not understand? Buji paisa tumange paisa? Thik ase. Okay, we have talked about plant breeding, we have talked about green revolution. Next topic ki asile? Biofortification. The next to it, another technique to enhance food is something called a single cell protein. What we understand? We say that it's not only these plants that they will always provide food for humanity. There are certain microorganisms also, there are certain unicellular organisms also which can be taken in as food. Right? Okol hey rice to khale je ami carbohydrates pam tenuka nohoy, okol je wheat e khabo lagibo ba okol plant product e khabo lagibo, not necessary. Kisuman microorganisms e, kisuman unicellular organisms e, single celled organisms bilakeo amak food diye, right? This is something that is included under this heading, that is single cell protein. For food, which type of single cell protein will you cultivate? Kunbilak bostu ami food hisabe use koru, right? Single cell or ki hobo? It will be certain types of algae, certain type of fungi. Before going forward, algae, one of the example we have already spoken about. Chlorella we have talked about, right? So there are certain examples of algae, certain examples of fungi, bacteria, etc. And obviously yeast, the most common example, right? So that is what is single cell protein. Single cell protein or contents khini podha. Just see if you have any doubts. And if you are talking about taking food from normal plants and taking food from single cell protein, which process is supposed to be more advantageous? It is obviously food from the single cell protein. Why? Karon normal food production jimla plants concerned hoy, they have a longer time for the production of food. Fertilization hobo lagibo, fruit develop koribo libo, pollination hobo libo, etc. etc. Right? Kintu single cell proteins bilakor duration kom samoyte ami khwa bostu tu pao, plus it requires lesser time, it requires lesser agricultural methods, and it is cost effective also. Right? Because of these reasons, single cell protein culture is more advantageous as a normal culture. Just read the part. Single cell protein or questions ki ahise? Kunbilak bostu ami single cell protein hisabe ami we can take it as food. Especially the examples of algae and yeast, Saccharomyces. Okay. So from food part, single cell protein we have completed. Swa, kiba doubts ase didi? Or else we move to the next topic, that is green manure. Scandesmus, SCENDESMUS, Scandesmus. Single cell protein bilakor ami ki koru, ami fermentation tankot grow koru. Bahut bilak examples di ase, but from examination point of view, it's generally the algae portion that has been asked, right? So, etu lesson basically mukhastor koribo laibo. It's not conceptual, eku bujiboleo nai. Okay. Doubts asil niki? Okay, then I think we can move to the next topic, that is green manure. First, what is manure? Manure mane ki? Fertilizers, right? If you talk about fertilizers, what is the image that comes to our brain? Certain types of chemicals that we use it as a fertilizer in the agricultural field. But it is not necessary that fertilizers or chemical components could only be used as manures or fertilizers. Kisuman natural fertilizers do thake, right? Those are called as green manure. For green manure, what you do? A green plant acting as a fertilizer is called as a green manure. Adult plant, hey plant dal jodi akko matit ami we sow it down, you have a plant growing in the soil, it dies off, it mixes with the soil and ultimately fertilizes the soil. That is what is called as the green fertilizer or the green manure. Is the concept clear? Green manure ba green fertilizer mane ki? Gos edal mati etat goji asile, he gos dal tate morile, tate mori pelai tate soiler lokot mili gol, ultimately ki korile he soil to tahate fertilize korile. So this is what we call as the natural fertilizer. Okay. Generally, which type of plants? Generally the legumes. Why legumes? Remember biological nitrogen fixation? We have talked about the development of root nodules, right? And if our plant has nodules, what it will have? Nitrogen content besi thakibo. So if the plant dies and it mixes to the soil and it already had a nodule, the entire content will go back to the soil and will ultimately fertilize the soil. So green manure ki hoy? Plants becoming fertilizers for other plant, that is called as a green manure. Which type of plant, to be more precise? It is the legumes that falls under this category. Not only legumes, beleg plant to ase, kintu common ki hoy? Legumes bilak. Some examples. Some of the examples you need to remember. Jeneke sun hemp. Both the two names, okay? Sun hemp question ahise. What is the scientific name? Crotalaria. Tar upor to question ahise. Question keneke ahise, example, the plant used as a green manure, right? So you need to know this. Then we have sweet cloves, dolichos, trifolium, etc., etc. Okay, the maximum question has been asked about crotalaria or sun hemp, and obviously about trifolium also. Legumes bilak kihor karone use koro? Because these plants are rich in nitrogenous compounds, right? Bracketed likha ase nodules are present. And these plants ultimately increases the nitrogenous content of the soil, it reduces the alkalinity of the soil and it prevents soil erosion. That's all. What is important? What is green manure? An example. Next we have certain plants called as energy crops. When we talk about energy crops, what we are talking about is certain plants that will produce alcohol, right? Those are called as energy crops. So kisuman plants or naam janibo leibo, from where we can extract alcohol. There are certain experiments that are conducted. Brazilot kori ase, USAt kori ase, ki kori ase. Enukwa plants kisuman bonabole chesta kori ase jar through tahate alcohol to deduct koribo pare, right? Some of the plants that gives alcohol are sugarcane, maize, potato, sugar beet, etc., etc. So these plants are called as energy crops. Question kiya hai, identify the energy crops. That's all. Next, petroleum plants. Petroleum plants, the plant from where you get exactly the same thing as, or same compound, exactly the same as petrol, right? Petrols bilak ki karone lage to running of automobiles. So if you can obtain that type of a chemical compound from the plant itself, hey plant to kami ki buli kom, petroleum plant buli kom. And whenever we talk about petroleum plant, generally one name that is asked is Jatropha. A Jatropha report ki question hai, belongs to which family? Euphorbiaceae family, right? So Tolotswa, you have certain families, Euphorbiaceae, Asclepiadaceae, Apocynaceae or Asteraceae. The plants belonging to these families generally could be considered as key plants, petroleum plants. So petroleum plants are what is important? The name of the families and the examples. Duta example diya ase, eta hol Euphorbia or eta hol Jatropha. Maximum question has been asked about these two only. Euphorbia, Jatropha. See up till here, is there any point that you did not understand? Tarpor alcohol bonuar process soli ase, experiment soli ase, and it has seen that it is quite successful. Eblack programs etiao pura release hua nai. You cannot be saying that sugarcane is the ultimate source of alcohol. Eblack experiment soli ase, kintu it could be extracted. Hekarne kisman program soli ase, soson Brazilot experiment soli ase, USA soli ase, right? So eblack plants or prao alcohol obtain koribo paribo, and later it could be commercialized. So yato hoi ase, yate khali amar naam tu nai aru. So just see up till here jodi kiba doubts ase, and we are left out with just the last portion of the lesson, that is tissue culture. Before going to that, ekhini kiba doubts ase niki swa. When it is in the form of a note, it is a huge note, but how you need to study or how you need to go for this chapter are the points that I have said. Baki bilak mukhasta nokoriba, it's just going to take your time away, right? So junkhini points mo mark kori disu ba important koso only hekiniye poriba, and rest of the thing you will have the idea from the MCQs. What is the question that is asked? There is no point of by hearting. PDF ot almost ticks in mara ase. Mara ase. Thik ase? Maximum ote mara ase. Molasses of sugar industry mane jeta sugar to bonua jay no, sugar to corpora bonua jay, sugar cane opera. So sugar cane opera jeta sugar to extract kora no, he process thake no, he sugar cane cane to ulabo tak tumi, you have to what, squeeze it down, tapra juice to ulabo leibo, thik ase, he rest over, he tare eta product ok ami ki boli ko, molasses boli ko. Thik ase? I think it's okay. Yato doubts thakibo nalage generally, right? Mukhosto koribo laibo upai nai. Okay, so the last thing that we will be studying is called as plant tissue culture. Plant tissue culture, the note is quite long, but questions ki aahe we will be discussing. Okay, first come to the word tissue culture. What is tissue culture? You culture a tissue, that is called as a tissue culture. What do you understand by the word culture? Division and okay, to some extent you are correct. Yes, there was someone who was asking me something. Kune khudi asile? Next page to repeat kori dise tar next page khonot, okay? Exactly repetition. Okay. Okay, so I was talking about something called as a tissue culture. What is tissue culture? You do the process of division and redivision of cells in a lab under certain nutrient medium, under certain environmental condition. That is what is called as tissue culture. You take something from a plant, you bring it to the lab, hey, eta cell ba duta cellor pra aru cells produce kora, that is what's called tissue culture, and ultimately from that collection of cells, you prepare whatever you desire. If you are talking about embryo development, you form an embryo. If you are talking about plant production, you form the entire plant, etc., etc. So this is what is called as tissue culture. First, tissue culture, natural and artificial, obviously artificial. What you do in tissue culture? You extract a cell or you extract a group of cells from a particular plant and then you culture or you do the experiment with. The part that we have collected from the plant has a particular name to it, and that is what is called as an explant. So question I have, what is the explant? The part that you have taken from one plant and you have cultured, that is called as the explant. Together with explant, we have something called as callus. So tissue culture পঢ়িলে দুটা word জানিব লাগিব, এটা হল explant আৰু এটা হল callus. What is explant? We understood. যোনটো part তুমি আনি পেলাই culture কৰিবা তাক আমি কি বুলি কম? Explant. Now what is callus? Callus, as the definition goes, no? কি বুলি কয়? Undifferentiated mass of cells is called as callus. Now what do you understand by that statement? Differentiation জানো নে নাজানো? জানিব লাগে কাৰণ আমি কথা পাতিছো differentiationৰ কথা. Differentiation, as of now, I think you should remember the zones of a root. We have talked about the zones of a root, no? So, eta area asile, eta kami ki boli kosilo, the zone of differentiation or zone of maturation. It is that area from where the root has developed, right? So, differentiation mane ki? What is the? I think we have explained this part before, na? Certain words like differentiation, de-differentiation, and redifferentiation. Ekhini ako porhim. কিন্তু তথাপি differentiation word to কলে what we understand? Differentiation মানে matured. When will you consider a particular group of cells to be matured? When it can do a particular function, right? Eta bostu differentiated conditionot ase means he is ready to perform that particular function. Liver, for example, our liver, no? Liver to kopra grow korse eta cellor pra. So, যেতিয়া এটা সেলৰ পৰা ডিভাইড কৰি পেলাই দুটা হল, দুটাৰ পৰা চাৰিটা হল, হেখিনি সময়লৈকে the liver did not conduct its function. কেতিয়া function conduct কৰিছে? Once it has grown into a particular shape, it has become matured, and only after that it has started working. So, that phase where an organism or a particular organ or a particular group of cells become matured and starts functioning, that is what is called as differentiation. Now, if differentiation is something that is called as matured and doing the function, what do you understand by the word called as undifferentiated? Mature one, functional one, right? Undifferentiated mass of cells. Eta cellor pra tissue culture korisa, ta mane eta cellor ami cell division korisu. Cell division kori pelai, suppose we want an embryo, we want to develop an embryo. Embryo develop koribo lagile, first explant to ami eta cell lobo lagibo na lage. Lagibo, hey cell to divide and redivide kori group of cells form koribo libo, hey group of cells to embryo বুলি কেতিয়া কম যেতিয়া ম্যাচিউৰড হ'ব যেতিয়া তাৰ পৰা বেবি প্লান্ট ওলাব তাকে আমি কি বুলি কম এমব্ৰায়ো যেতিয়ালৈকে আনডিফাৰেন্সিয়েটেড কন্ডিশন হৈ থাকে যেতিয়ালৈকে ম্যাচিউৰড নহয় সেই গ্ৰুপ অফ সেলস ইজ কল্ড এজ হোৱাট ক্যালাস এটা সেলৰ পৰা ডিভাইড কৰি কৰি এমব্ৰায়ো ফৰ্ম কৰিছে এমব্ৰায়ো কেতিয়া কম যেতিয়া হি ম্যাচিউৰড হ'ব the group of cells should become matured should be able to function that is what is called as differentiated condition up till the time it is matured that stage is called as what callus right so tissue culture duta word eta hol explant aru eta callus tissue culture code koru labs generally if you want to make a cell divide you have to put the cell or you have to give the cell certain types of food to kami ki boli call nutrient medium right tissue culture always requires nutrient medium and if you talk about nutrient medium not necessary it will be always solid it could be liquid also so liquid nutrient medium o ase solid nutrient liquid medium o ase so let's see it is a technique of growing cells tissues organs or organisms in sterilized nutrient medium under aseptic condition obviously aseptic ba microorganism free hoboi lagibo nohole ami chuntu plant grow kori aso tak dohon already microorganisms infect kori dibo so the experiment that we are conducting in that area should be microorganisms free aseptic right okay next could be cultured in vitro in vitro janu no away from the body that's called as in vitro okay the technique of tissue culture was first started by question has been asked about that so scientist name important Haberland right For tissue culture, you need to apply certain types of hormones. Which hormones? Auxin, gibberellin, abscisic acid, etc., right? If you have read the hormones part, auxin, I think you all remember, auxin কিহৰ কাৰণে use কৰে? To increase the shoot apex and the root apex, right? To increase the girth, you apply what? Gibberellins. Internodal elongation, gibberellin. As a whole, cell division, cytokinin, right? Okay. So tissue culture কি লাগিব? Aseptic conditions লাগিব, nutrient medium লাগিব, আৰু hormones লাগিব, right? So what are the essential requirements? Nutrient medium, aseptic conditions, and hormones. Talking about nutrient medium, as I said, it could be solid and liquid. One example of a common nutrient medium is MS medium. So question to ahe, what is the most common nutrient medium used during tissue culture? It is MS medium. Full form of MS medium diya ase molasses and slug medium. Components of the medium nalage, it's not required. And how will you make the condition aseptic? That diya ase, right? Antibacterial, antiviral, chemicals কিছুমান use কৰিব লাগিব, boiling, sterilization, এইবিলাক methods apply কৰিব লাগিব, and ultimately we do what? Aseptic conditions. Nutrient medium consists of what? It consists of inorganic salts, a source of carbon, amino acid, and growth regulators. Iman details or question aha nai. Khali tissue culture ki lage? Nutrient medium, aseptic conditions, and certain types of hormones. This is something that we are going to study. Next, the terms are important. Just go through the terms. We have a word called as micropropagation, something called as a Soma clone, and the third is subculturing. Please go through these three words and see whether you have understood or not. Micropropagation mane ki, Soma clone mane ki, aru subculture mane ki. Question he bilakor upor ahe. Auxin. Abscisic acid. Abscisic acid কিছুমানৰ particular normally to আমাক gibberellin আৰু auxin বা cytokinin এই তিনটাই লাগে কেতিয়াবা in certain cases এটা part of the tissue you don't want to develop ঠিক আছে you don't want হে callus ৰ পৰা এটা part তুমি কাটি দিব খুজিছা so in that case you could be using certain other types of hormones also কিন্তু mainly কি লাগে gibberellins auxins আৰু cytokinin নহয় নাই নাই ক্ষতিকাৰকৰ কথা নহয় কেতিয়াবা তাক আমি ইউজফুল পাৰপাসৰ কাৰণেও আমি ইউজ কৰিব পাৰো because this is everything that is man made natural হলে বেলেগ হ'ব আৰু artificial হলে বেলেগ হ'ব আমি নেগেটিভ এসপেক্ট এটা পজিটিভো কৰি লব পাৰো ওকে ওকে যিকোনো প্ল্যান্টৰে আমি জেনাৰেলি টিস্যু কালচাৰ কৰিব পাৰো okay okay so the first callus yes prepared phase to hey cells or collection kinik যেতিয়ালৈকে তাহাঁতে ম্যাচিউৰড হোৱা নাই তাহাঁতে কাম নকৰে ন ম্যাচিউৰড হোৱাৰ আগৰ ষ্টেজত আমি কি পাম কিছুমান সেলছৰ কালেকশন পাম সেই সেলছৰ কালেকশনটো আমি কি বুলি কৈছো from tissue culture point of view the word that we use is called as callus ঠিক আছে এটা সেলৰ পৰা ডিভাইড কৰি গ্ৰুপ অফ সেলস ফৰ্ম কৰিব সেই গ্ৰুপ অফ সেলস টু না তুমি স্টেম বুলি ক'ব পাৰিবা না তুমি ৰুট বুলি ক'ব পাৰিবা now you can consider that group to be a embryo now you could consider that to be a fruit etc to কি বুলি ক'বা we'll be using a word called as callus callus or pora ultimately differentiate বা mature হৈ পেলাই respective parts বিলাক form কৰে clear okay what is micropropagation it is the technique of making many plants by the process of tissue culture the process is called as micropropagation propagation mane ki reproduction micropropagation you started from a single cell so that process is called as micropropagation next we have something called as a Soma clone what is a Soma clone each plant produced by tissue culture is called as a Soma clone right micropropagation বা tissue culture or karone but through re আমি যিমানবিলাক plants produce কৰিছো সেইবিলাকটো sexual reproduction নহয়. So that is the means of what? Asexual reproduction. So if it is asexual reproduction, each and every organism will almost look the same. Almost key, it will look the same itself. So each and every one could be considered as a clone, and in tissue culture language we use a word that is called as a Soma clone, right? Next, subculturing. What is subculturing? We have been using a word called as culture. What is culture? You take a cell, you make the cell divide and redivide, that is called as culture, right? What is subculturing? So when you talk about tissue culture, if you are talking about a cell that is undergoing tissue culture or the cell taking in as an explant, what will you do? Eta cell loba, eta petri dish loba. In that petri dish, you will put certain types of nutrient medium and you put the cell. For example, moi ki koiso je during the process, the most common type of tissue culture, you have a petri dish. In the petri dish, what you have is nutrient medium or the food, right? And the food could be solid or liquid, we understand, right? In this nutrient medium or in this food, you put a cell, and this cell will be cultured. Cultured means you will make the cell divide and redivide, right? So after that, ki thakibo? This is a petri dish, this is the food, and ultimately, gradually, what you will see? The cells has increased. So this is what exactly is happening is culture. Once if you leave this like this, ki hobo? If the cells keep on increasing, obviously ki hobo? Yar nutrients to komi jabon na jay. The food quantity will decrease. If the food quantity decreases, and after that also you are leaving the cells like this, what will happen to the cells? They will get hampered because of lack of food. So what you do is, you take out certain cells from this, right, from this part, দুটা তিনটা এনেকে space কৰি দিবা, you shift it to another tissue culture with, sorry, you shift it to another petri dish, right, with again nutrient medium. This is called as subculture. First to ki koisilo? Culture. প্ৰথমতে কি কৰিবা, যোনখন পেট্ৰি ডিছত ফাৰ্ষ্ট কৰিছো, সেলছবিলাক বাঢ়ি বাঢ়ি গৈ আছে, সেই প্ৰচেছটোক আমি কি কৈছো? কালচাৰ. কালচাৰৰ কাৰণে কি হৈছে? দ্য cells kept on increasing. If these cells keeps on increasing and the food keeps on decreasing, there might be chances that the cells will die. So what you do? You separate the cells. Belek belek petri dishot thoi dia, that is what is called as subculturing, right? Just go through the sentence whether it's clear or not. Subculture. So, eta cell ase, cell edal plant ase, thik ase. Suppose we have a plant, thik ase. A planter eta cell ami collect koriso. A cell to ami ki boli kom? Explant. A explant dal ami ki korim? We have brought it to the lab. In the lab, we have taken a dish. A dish khanor naam ki? Petri dish. Petri dishot ebilak ki? Food. Foodok ami ki boli koiso? Nutrient medium. Nutrient medium thaka ekhon dishot a explant to ami thoi dilu, right? With proper everything, no, hormones, chemicals, whatever it is necessary. What we'll do? We will allow this cell to divide and redivide. Eto cell e jete khabo paise he divide koribo nokore? Divide koribo. So divide jodi kori thake, prothomote eta cell asil, etia iman bilak cells ase, ei khwa bostu to komi jabon na jay. Jedi khwa bostu to komi jay, gutei kita cell affected hobo nohoy. Affected hoi pelai to moriu jabo pare, no, because it suffers from something called as lack of food. So what we do is, a gutei kita, suppose yare 10ta cell asile, no, tarpor ami 5ta cell yale loi anu belek ekhon dishole, to yate coming golle nai. এইটো প্ৰসেছক আমি কি কম? সাবকালচাৰ. প্ৰথমতে কালচাৰ কৰিলো, তাৰ পৰা আধা আধা সেপাৰেট কৰিলো, এই সেপাৰেট প্ৰসেছটোক আমি কি কৈছো? সাবকালচাৰ. সাবকালচাৰ কিহৰ কাৰণে কৰো? So that the cells grow properly and healthy. और nutrients দিম, নতুন পেট্ৰি ডিশত আকৌ নতুনকে নিউট্ৰিয়েন্ট দি পেলাই আকৌ গ্ৰো কৰিম. Clear? And the last part is types of tissue culture. What are the explants that you take, right? So first is callus culture. Swa. Culture medium. What is the culture medium that you use for callus culture? Callus culture mane? Types of tissue culture. The first one is callus culture. What is callus culture? From one cell, you want to grow a group of undifferentiated cells, that is called as callus culture. Where will you do? You will do it in the lab. What will you use? You will use certain types of nutrient medium, right? As far as the nutrient medium that is used for callus culture is concerned, what is the common nutrient medium that you use is agar-agar. Remember agar-agar we have talked about, no? Kod porisilo? Algae, red algae. So what you do is in this process is you take a small piece of a parenchymatous tissues, introduce that into a culture medium, give the hormones, etc., and ultimately you get a group of cells that is called as callus. This process is called as callus culture, and from that callus you can develop any part that you want, right? Tomar jodi okol suppose eta cell ase, thik ase, you have one cell and from here what you have developed is a group of cells, right? This is called as what? Callus, and from this group you want to develop only the shoot of a plant, right? Root dal amak nalage okol shoot lage, because obviously from plant point of view सबसे important ki shoot e hobo, right? A callus khini pra enukwa eta condition korim jot niki okol shoot khini grow kore, karon root would be what? Unwanted parts, right? So ami ki korim certain types of chemicals dim jar karone yar pra okol shoot se develop kore, right? This is what is called as tissue culture. Chemicals di pelai ki koribo nwari, sobe koribo pari. Gosdalok root kiyo lage? Why does a plant require root? Matir pora. To ami gosdalok to matir ruai nai no? To root dalor kam ki? Hoine? Tai hi junbilak minerals matir pora loy, hebilak minerals jodi ami artificially di diu, eketa kotha hol ne nai? কিয় হেৰিত নাথাকিব নেকি, জাইলেম আৰু ফ্লোয়েম নাথাকিব নেকি ষ্টেমৰ ভিতৰত? ভাস্কুলাৰ বান্ডলছটো থাকিবই, খালি সি নিজে কামটো কৰিব নালাগে, মই কামটো কৰি দিছোঁ। আমি খোৱা বস্তু নিজে বনোৱা নাই, ছুইগিৰ পৰা অৰ্ডাৰ কৰিছোঁ। বুজি পাইছা? কিন্তু দ্য মেইন পইণ্ট ইজ হোৱাট? নিজে বনাই পেলাইও খাইছোঁ, ছুইগিৰ পৰা আনি পেলাইও খাইছোঁ। য়েছ অৰ নো? So we can do anything. We can do anything. Okay, chalo. Okay, read the next part. Multiple shoot production. So you have a callus. From this callus, you want to develop many shoots. That is what is called as multiple shoot production. So ki dibo laibo? Chemicals to obviously dibo laibo. So what's the important chemical ki? Hormones. Hormones dilehe shoot develop koribo. हाँ हेतु तो नेक्स्ट टाइम আছে हाँ बेसिके ऑक्सिन दिले সদায় रूट फॉर्म হয় बेसिके साइटोकाइनिन दिले हेरी फॉर्म হয় शूट फॉर्म হয় एटु এটা very commonly asked question from tissue culture. High auxin is to cytokinin ratio and high cytokinin is to auxin ratio develops what, right? As a next thought swa. The number 2 is suspension culture. First ami ki porilo callus culture, second is suspension culture. What is suspension culture? It is also a type of tissue culture. This time the nutrient medium is not solid but it's a liquid nutrient medium. Callus culture ki asile solid nutrient medium. Suspension culture ki ase liquid nutrient medium, right? Number 3, shoot culture and root culture. From a cell you have developed callus. From the callus, if you want only the shoot to develop, what you need to give is a particular hormone. If you want only the roots to develop, then also you need to give a particular hormone, or the concentration of that hormone should be more as compared to the other hormone. Whenever we are talking about divisions or we are talking about tissue culture, the most hormones that are associated is obviously auxin and cytokinin. So sometimes if you increase auxin ratio as compared to cytokinin, or auxin amount as compared to cytokinin, kiba eta to hobo, right? So normally ki lage auxin or cytokinin এটা particular ratio lage, ঠিক আছে? Auxin to basically di dile কিবা এটা হ্যাম্পাৰ হ'ব অক্সিজেন বেছিকৈ দিলে কি হ'ব প্ল্যান্টডালৰ ৰুট সিস্টেম বেছি ভালকৈ ডেভেলপ কৰিব সাইটোকাইনিন বেছিকৈ দিলে কি হ'ব শুট সিস্টেম যদি অক্সিজেন আৰু সাইটোকাইনিন দুইটা একে সমান দি দিয়া কি হ'ব equal amount of oxygen and equal amount of cytokinin would result into what callus এইটো বস্তু আছে তোমালোকৰ আৰু এটা লেসন মই পিছত পঢ়াম ঠিক আছে হৰমোনছ পাৰ্টত আছে খালি এতিয়া কি মনত ৰাখিবা more oxygen than cytokinin induces dash induces root more cytokinin than oxygen will induce the shoot so callus of cytokinin basically shoot hobo oxygen basically root hobo equal concentration neither shoot nor root so that group would be called as what callus right oh generally hoy kintu tumi lab conditions of koribo lagibo it is not only the hormones that are responsible aru beleg bostu responsible chemicals u lagibo okay so the next is somatic embryo culture etu section ot ami ki pori aso je tissue culture ami ki kori aso we can go for something called as a callus culture we can go for suspension culture we can go for root tip culture or shoot tip culture jikono bostu ami nije banabo paru right next is somatic embryo culture somatic embryo mane ki what is body cells of embryo form kore niki who forms the embryo it's the body cells or it is the reproductive cell so generally actually no hoy commonly it is sexual reproduction hole it is always the germ cells that produces the embryo after fertilization but you want to make an embryo from the body cells so since we are doing it artificially this process would be called as what tissue culture and for to do that process obviously ami ki dibo laibo chemicals dibo laibo right so somatic embryo culture is one method of tissue culture jot ami ki koru somatic cells opra embryo banao নহয় solid to tahator kisuman chemicals or through re tak solubilize kori pelai dibo laibo kintu first use korute itu solid asile kintu suspension culture first use korute liquid asile okay okay so that is somatic embryo culture next we have embryo culture means you have one embryo you want to make many embryos from that one embryo that is called as embryo culture what will you do you will take the embryo the normal embryo somatic embryo no hoy gametic embryo okay you take the embryo cut the embryo into pieces develop each piece into the respective embryo eta embryo pra bahut bilak embryo form korila by the process of splitting or cutting or incision this is what is called as embryo culture eta embryo pra tukura bilak loi pele aru embryo form korilo ha please say naturally keneke koriba polyembryony is a soap plants at hobo not necessary ami embryo kihor karone banaisu so that we can have a baby plant there are certain plants where the embryo doesn't develop properly remember we have talked about orchids or seed or pra orchid plant edal hoy janu no hoy kyo no hoy because it does not have the food where is the food found obviously in the embryo so ami jodi enuka ta embryo form koribo paru in case of a orchid jot food ase te tar pora to plant edal hobo right so ebilak culture what we use for our benefit right so you make the embryo by incision of the young embryo this is called as embryo culture it is applied in or this process is used for something called as embryo rescue so asun tolo paragraph to embryo rescue kotha dia ase what is embryo rescue rescue mane save right Why do you have to save the embryo? From whom? Predator prey to nai that you have to save the embryo. So generally we use this term in case of tat likha ase swa interspecific hybridization. Interspecific hybridization mane hetu to intra hobo no. Interspecific, two different plants, two different animals, eta dog aru eta cat duita fertilize korile offspring to বা embryo to nathakibo to pare moriu to jabo pare. In majority of the cases they will become sterile, right, or they will die. In case of plants, if two different plants, tomato and chili, you mix it up, the new offspring, obviously the embryo will die, right. So you want to save that embryo, so tetia ami ki koru, we go for methods of tissue culture, right. That is what is called as embryo rescue, generally done in interspecific hybridization. And whenever you talk about embryo culture, we generally remember the example of orchid. Next, another part that could be cultured is anther. Anther culture. Anther culture mane ki? Eta anther opera plant you will make, right? If you make a plant from an anther, what would be the ploidy of the new plant? Haploid, right? Mujhe paisa na? You have a plant. The plant has a male reproductive part. The male reproductive part obviously have the pollen grains. The pollen grains need to get pollinate, it has to undergo sexual reproduction, then only a new plant will be formed. It is going to take a longer time. Instead of that, if you can just collect the pollen grains and make a new plant, that will save your time, it will go for faster reproduction, faster production of plants. So from certain anthers also we can make a plant. Kintu plant to initial stage of ki hobo? Haploid. This haploid condition we have to make it diploid by application of what? Colchicine. Remember we have talked about colchicine. Colchicine ki kore? Doubling of the chromosomes. Remember cell cycle and cell division on koisilo no? Metaphase stage of the colchicine di dia tete hale ki hobo? Chromosomes bilak separate no hoy. So if it doesn't separate, obviously the chromosome number will keep on increasing. So doubling of chromosome, one chemical that is used is obviously colchicine. So if you go for anther culture opera jundal plant hobo, first it will become a haploid. You apply colchicine, make it diploid, and you make it a normal plant, right? These are all methods of anther culture. Okay. Colchicine metaphase or delay anaphase to bond hoi jay, anaphase na jay goi. Dekha se equatorial planote thaki jay. Okay. The next type is meristem culture. Why do you culture a meristem? First, what is a meristem? Meristem ki hoy? The part that has the meristematic tissues is called as a meristem. Why do you go for a culture of the meristem? Culture, why do you want to increase the number of meristematic tissues? Because meristematic tissues are supposed to be virus free, right? In the entire plant body, jiman bilak cells ase, gutei cells bilakot ki hobo pare? Infected hobo pare. There is a chance. Why? Because they do not divide. They have a resting phase, they are permanent, so there is every chance that the virus and the other microorganisms would attack. But if you are talking about meristematic tissues, they are in a continuous state of division. So imane active that tahatok infect kora, it's very hard. So generally the meristem of a plant is virus free, and if you want to develop a new plant that is virus free, what will you culture? It is always those tissues that you culture. Kunbila tissues jar virus se infect koribo nware, and which tissues? Meristem. So this type is called as a meristem culture. Why do you go for meristem culture? To develop a virus free or disease free plant. Yeah, ask. Oh, soap plants hote to meristem. Soap plants hot kyo grow koribo nwara? Soap plants hot meristem na thake niki? Meristematic tissues plants hot na thake niki? Oh, heidal plantor tumi jodi meristematic tissues ani pelai tenekwa dhornor edal plant bonai dia, hei plant dalor aru disease nohoy generally. disease resistant. So disease resistant generally kunbilak cells or karone ami heri koru, it's always the meristematic tissues because they are in a fast active mode of cell division. Okay. So question ahe, virus free plants produce koribo lagile, which part will you culture? You will culture the meristematic tissues or meristem, right? And finally, the last part of culture is the protoplast culture. Protoplast culture par eta question ahise, what is a hybrid? I think you have heard the word called as hybrid. Hybrid shunisan nai? Hybridization but tissue culture kole ami hybrid jano. What is a hybrid? You cross two different plants, the new offspring is called as a hybrid. Now we come across a term that is called as a hybrid. So question ahe, what is a hybrid? Hybrid ki hoy swasun? Likha ase. Okay. What is a hybrid? Read the line, see the diagram. Is it understood? First, hybrid is a product of protoplast culture. Protoplast culture mane, what is protoplast or protoplasm? The content inside the cell wall or the cell membrane that is generally referred to as protoplast. So for protoplast culture, what you do? You take two cells, you fuse them, and in that stage, ki paba? So if you are talking about cell A and cell B, jodi fuse kori dia, you get two conditions. You get a bigger cell, tat ki thakibo? A2 cellular protoplasm fuse hoi thakibo, plus this nucleus and this nucleus will fuse together and will form a bigger nucleus. This might be the condition, or it could be a condition যে ইয়াৰ protoplasm fuse হৈ গল কিন্তু nucleus kita fuse নহল. Are you understanding? So for protoplast culture, what you do is you take two cells and you fuse them. If you fuse two different cells, there might be two conditions. One, in a condition where everything fuses, nucleic material fuse korile, protoplast fuse korile. The other where only the protoplast has fuses, right? Yate ki hoise nuclei duta beleg ke ase. So out of these two nuclei, if one nuclei degenerates, this cell would be called as ki? Hybrid. Now read the sentence that is given in your PDF. Protoplast culture is also called as somatic hybridization. The term is important. Hybrid of part 2 pora, and I think this is all for today, and this is the last end of this topic also. Example diya asa pomato and bomato hegita saba potato ha তেতিয়াহ'লে normal না তেতিয়াহ'লে normal condition হ'ব হেডাল প্লান্টক আমি hybrid নকওঁ hybrid ক'ব লাগিলে এটা condition লাগিব সেই conditionটো যদি হৈছে তেতিয়াহে তাক আমি hybrid কম আৰু hybrid নোৱাৰে fuse যদি হয় হেতু সেলক আমি hybrid নকওঁ এটো তো hybrid হৈ যাব hybrid হ'ব নহয় এটো এটা সেল দুটা সেল fuse হৈ পেলাই product form কৰিছে এটোক আমি কি বুলি কম hybrid কিন্তু এটোক আমি কি কৈছো hybrid chemicals or through korabo para artificial keteba nije natural conditions or karne hobo pare generally if you are going for this one na if you are going for tissue culture this is something that is done artificially so ami degenerate korai diu degenerate korile ki hobo protoplast khini contents besi thakibo maybe that is of much more economic importance as of the genetic material আমাক যদি protoplasm বেছি লাগে protoplasm qualities খিনি বেছি লাগে এডাল প্ল্যান্টত তেতিয়াহলে protoplasm বেছিকে থাকা আমি সেল এটা কালচাৰ কৰিম protoplasm বেছিকে ক'ত থাকিব হাইব্ৰিডত okay so I think this is all for today so in this entire chapter what you do is just by heart right so what is important plant breeding or points kita saba next we have talked about what green revolution or gene naam gita father of green revolution semi dwarf variety right and variety of wheat variety of rice identify jaya ratna sonalika kalyan sola larma rosa next biofortification duta birat important line first is legumes slacken or cereals slacken and what is biofortification and why do you go for biofortification to increase the protein the vitamin the mineral etc contained in a plant we go for biofortification next energy crops it's just the example that you remember right generally sugarcane etc from where you can obtain alcohol next is petroleum crop it is generally the family Euphorbiaceae you remember question ahise maximum petroleum crops are obtained from which family Euphorbiaceae jedi Euphorbiaceae nai Asclepiadaceae thakibo right by Asteraceae thakibo best example Jatropha and Euphorbia just two examples after that what is left with single cell protein the examples of algae that is most asked right and yeast lastly we have tissue culture point of terms definitions black zanibole what is callus culture what is Anther culture right question has been asked about what is a hybrid etc right and obviously table 9.1 and 9.2 right thank you that's all for today